panel. And our panel is going to be on structure performance processing in fungal biofabrication and fungal biomaterials. Tomasz is remaining with us for this panel. Thank you, Tomasz. And we're joined by two colleagues here from Penn State University. Professor Felecia Davis is Associate Professor of Architecture and the Carey Memorial Early Career Professor in the Arts here at Penn State. Welcome, Felecia. And Professor Josephine is Assistant Professor of Food Science, also here at Penn State. Welcome, Josephine. Thank you both for joining us. And like I said, thanks, Tomasz, for staying with us. I did want to give you just a very brief introduction as to our goals for this panel, and we have two goals. First, we want to share state-of-the-art in fungal biomaterials as related to engineering, architecture, construction, and design. And our second goal is to inform you that issues related to growing, extraction, processing, structure, property, performance, all require understanding of biology and chemistry and types of fungal biomaterials. One of these were questions this morning that we posed, and in particular, contamination issues and interaction with other materials. So we'll talk about other related issues as well, but these are two main goals that we use to put together a list of prepared questions, but we will also pause and take questions from the in-person audience as well as the audience on Zoom. So first, we're gonna hear a little more from the panelists, in particular, both Felecia and Josephine, who've just joined us. Then we're gonna go to a couple or a few of the questions that we prepared, and we will pause to take questions from you here and on Zoom. Okay? Are we ready to go, panel? Sounds like we are. Okay. All right. Felecia, may I ask you to get us started with some introductions, and in particular, your research expertise related to fungi and biomaterials?
Sure. Can you hear me? Okay, great. All right, thumbs up. So thank you, Zoubeida and Benai, for the invitation to come speak on this panel today. I did prepare some slides to share with everybody by way of introduction, and to talk a little bit about the work that I'm doing with Benai and others in the SEDC. So is it possible to share those? Looks like. Okay. So I'm hoping everybody can see that because I can't see anything else. So this is really, really quick. It's a capsule of work that I am working on with Benai, Ali Ghazvinian, and also Farzana Aghazian, students in Format Lab and Soft Lab. And it's brand new territory for me. I am the director of Soft Lab, which you can see here, which is in the Strickman Center for Design Computing. And there we have all sorts of equipment for dealing with textiles. So I really work a lot with different kind of ancient even patterns and making of textiles. And we primarily work on textiles that respond to their environment via programming embedded sensors and using the natural responsive properties of fibers themselves, where the response is used by the designer to communicate some type of information to people. So that is primarily the kind of research agenda in my lab. But there are two different scales that I have been working on that agenda in the School of Architecture. One is more architecturally traditional, which you see before you, which are things like tents, shade structures, etc. And the other direction is related to things that you wear in your body to augment the body so that we can begin to be sensed and sense our environment around us. So today I really just want to share with you very briefly Microknit, which is a building structure that we're going to develop. And it's a fiber composite made of a knitted base with mycelium embedded into it. And it is also a kind of interdisciplinary project. We're working with someone, Andre West in North Carolina State in the knitting lab there, as well as John Pettia, who is from the Department of Plant Pathology and Environmental Microbiology here at Penn State. So we're a pretty multidisciplinary team. And the project is really to do a 1-year long project to construct a full-size shelter using CNC knitted technology embedded with mycelium fungus. And it's been awarded a prize by the SOM Foundation in Chicago. And the shelter, or this pavilion, will be constructed during a directed research studio in the fall of 2022 within the Department of Architecture. So we're going to have students working with us, both upper level undergraduate students and Master of Science students working together to make this structure. So one of the critical issues that we're dealing with in architecture is scaling up. And what you can see here is, a couple of small samples. I think these are no larger than about 8 inches or so. And we've been really trying to figure out how to learn from feedback loops between these different scales, right, in terms of designing the material. So we're taking a multi-scalar approach to our project and starting at a couple of different places along the scale continuum. So this is a very recent shot of a hyperbolic paraboloid model that we have in John Pettia's mushroom growing lab. And this is before we put mycelium on it. And then you can see another shot of it, and you can get a sense of how big it is. And here is Ali, another co-PI on this project, applying the mycelium to it. And what happened was we lost our double curve. We had a beautiful hyperbolic paraboloid that, once it got weighted down with the mycelium, changed its shape. And so we've been also looking at working with predicting shape legibility with this project. Farzana Aghazian has spearheaded that, and she's been working on that with knitted fabrics in particular. And right now we're in the middle of this project, and some of our research questions, just to close, are how to deal with the stretch and change of the textile with the addition of the water from the mycelium, the best ways to add the mycelium, building, should we build with contamination or without it? And we have a health and safety question there. And personally, something that I'm very interested in is, at what stage can this process or this material be brought outside the lab so that people could potentially work with it to make their own low-cost structures? And is the way that we should think about this the way, similar to making a brick, where you make the brick and then you provide the bricks to people? Or is it a process that the whole process that we can take outside of the lab and allow, for example, collective building to happen? So I have many more questions, and my teammates do as well, but I'll stop with that. Thank you.
Great introduction, Felecia. Thank you. Josephine, may I ask you to jump in next?
Sure, yeah. Okay. Can you hear me okay? Good.
Yes.
Okay, perfect. I also have a couple slides that I put together for today's panel on structure, performance, and processing. So just a little background on myself. I'm originally from Malaysia. So just to kind of frame where I'm coming from in terms of this conversation on biomaterials from fungi. So I did my bachelor's in food science and PhD in food science and toxicology. So I come from a lens of also safety and quality here in this conversation on using fungal biomaterials. And then I went on to do a postdoc at Cornell studying evolution of genes. So just to take scales down a couple scales smaller, I'm a microbiologist by training. So I think about how we can change the fungi or work with the fungi to build these materials that we're trying to get to. And then I started my lab here in 2018. So what you see here, we're housed in the food science department. We're on the 4th floor here because as you have similar issues with contamination, our microbiologists are kept on the 4th floor, high and far away from our creamery and our food operations. Okay. So this is one slide to summarize our lab. This is how I think of the world of fungi. Our lab is interested in this connection between genetic differences and phenotypic consequences. And what do I mean by this? Like I mentioned, we're really interested in genetic structure, so changes in genes throughout evolution that can select for or select against specific phenotypes, okay? So what you're seeing here at the bottom here are two pictures of they're scanning electron micrographs, so they're microscopy images of two very different fungi. And we have questions on porosity and the architecture of the mycelium itself that can select for specific properties that we want in these biomaterials. And I have a last slide here that, oh, okay, let me add, so some of the skills that we have in our group, we think about these approaches from a microbiology standpoint. So we optimize fungal physiology. So all we think about here is from the fungal perspective. So we have experience in molecular cloning, protein expression, as well as strain engineering. So for those of you that are interested to sort of collaborate in this space, this is how we envision our role in helping advance the field of fungal biomaterials. And this is just a zoom in image of what I just showed you. So again, we're interested in what are the genes that drive mycelium morphology and architecture. So on your left here is mycelium obtained from Rhizopus oligosporus. So Rhizopus is a food mold, right? So this has been used for centuries in tempeh production. So tempeh is somewhat a food composite material. So that's how I think about molds and fungal biomaterials. So you basically inoculate or grow Rhizopus oligosporus with soybeans and grow a composite. So I think about it from a food perspective. And then on the right here, you see Phanerochetes chrysosporium. Sorry, I think I missed a Y there in my spelling. Phanerochetes is a white rot fungi. So again, these are taken at very similar scales at 100 microns, but their evolutionary morphology looks so different. So one of the things that we're really interested in is how can we select for these genes to give us a better idea on which types of fungal strains we can select for building of these different materials. So Zoubeida, I'll turn it back to you. I'll stop my share here.
That was great, Josephine. Thank you. Tomasz, did you want to add any introductions for yourself?
No, I think I have said everything I have to say. Thank you.
Okay. Well, hopefully you'll have more words for the
yeah, some words left, definitely. Yeah.
Yes, thank you. Well, that was great. So what I'd like to start is with a question that is going to be sort of general, but I wanted to build on what we learned this morning, for example, right, through the talks. And so the question is, and this is to all three of you panelists, how do we define fungi as a biomaterial? And what are attributes, chemical, structural, others, that make them attractive in contrast to synthetic materials? Tomasz, did you want to get us started, and then we can get to Felecia?
Yeah, I think it's like with all bio-based materials, they are easier to compost, so they are easier to feed back in a sustainable loop. They can biodegrade after use, and I think the mycelia, they are really interesting because they make a complex 3D network which can be used to stabilize complex structures at the end. We have seen it by Felicia's clothing and stuff like that. You can more or less put fungi nearly on everything as a scaffold, and then the fungi makes their trick. I have to admit, maybe it's a German point of view. We have strict regulations how to use fungi in contact with humans, and therefore we had, before we are allowed to test it in our labs, we had to put them in an oven for 120 degrees for 2 or 3 hours. And I think for architecturing applications of fungi, one has really to keep in mind to make this absolutely safe. But I think Felecia and Josephine can say much more on this issue from the point of view of, as a mycologist or as a one who works with fungi and clothing.
Yes, I think that's exactly right. I think we have similar regulations in the United States applying fungi. As a matter of fact, as an architect, if there is fungi in your building, you have some kind of failure, right?
Yeah.
It's a big mistake. And so, yes, this is something to definitely overcome, I think, to take the fungi to architecture. The other thing I wanted to say in terms of its usefulness is that, you know, I think 40% of landfill comes from the building industry. We have a really scandalously high input into the landfill situation. And so using a material like this helps reduce that problem.
And I can jump in here. So going back to the question, I think I approached this from two ways. So the first question is, how do we define fungal biomaterial? So fungal mycelium on itself is actually very brittle. So if you look at the data on mechanical strength, on stress and strain curves, pure mycelium has horrible, almost garbage, my graduate student puts it that way, horrible mechanical properties, you know? So when he first got on this project, I asked him to put together, you know, what are these fungal mycelium? And his first slide was, pure mycelium has horrible mechanical properties. Okay, so we know that we cannot build structures just solely on pure mycelium. We need substrates. So then we go into ag waste, for example, agricultural waste, brewer spent grains. So we use these substrates to coax the mycelium to bind these substrates, complex substrates. So actually the performance comes from the substrates in which we choose as much as the fungal strains that we select. So I think that is the first way how I define fungal biomaterials. So I think about it from a food perspective. So you think of your beautiful camembert cheeses of your world, right? Penicillium roqueforti, for example. Outside, it's covered by this beautiful, delicious white fungi, but on the inside, it's actually a dairy substrate, right? So on itself, fungal mycelium cannot perform on its own. We need a substrate, and the substrate determines its final mechanical strength. And then the second question that I'd like to address that Zoubeida asked is, how are these natural materials better or comparable to materials? And I think just looking on pure mycelium, they are sustainable, but the issue comes with the post-processing steps that go with it. That now, because these mycelium have weak mechanical properties, we have to process them in a way that increases their mechanical strength, but at the same time decreases their sustainability. So we think about this from a food perspective as well. How do we scale? And I think the lessons we can borrow from food science is that we can produce food with fungi on a large scale with cheap resources, right? So I think there's a lot we can borrow from this and in food science to apply to fungal biomaterials. Zoubeida, I'll turn it back to you.
You brought up some really excellent points, Josephine, that we're going to want to revisit. Let us go to Zoom, and I saw a couple of questions that are very relevant to you know, where we got to this point.
Yeah. So one question is about the pure mycelium, Josephine, that you mentioned. The question is, if you dry pure mycelium, can it become a hard foam with some mechanical strength, or is it still terribly weak?
Yeah, that's a great question, Michael. Michael asked a question, you know, if we dry it, is it still terribly weak? Drying actually decreases the mechanical strength of the mycelium, but it depends highly on the strain. So just taking those two different fungi that I showed you with Rhizopus, it's very brittle, and Phanerochetes sheets chrysosporum, if you up its chrysosporum, it's much more stronger. It's stronger than Rhizopus. So there is something inherent in the structure of the cell wall that selects for this mechanical properties. So for now, we're seeing that drying actually selects against high mechanical strength.
Josephine, you just made an interesting point, and there was a question this morning from Maria, actually, I'm not sure if she's still here. She asked about strains of fungi and why we use what we use. In particular, you know, it seems like in the community that uses fungi, there's maybe 4 or 5 types that are used versus, I don't know how many, more than thousands, right? Types are available, millions are available. And you made a point in terms
Millions,
of
yeah,
attributes.
millions are available.
Millions are available. You made a point in terms of attributes. Can you revisit that and tell us maybe a little more as engineers and scientists? Why is it, and artists and architects, why do we use some and not others?
Sure, thank you, Zoubeida. So I'm just going to repeat that question from my perspective. So your question is, why do we use selected strains like Pluralis and Ganoderma to make these biomaterials? So I think the one thing that we really can tap into is evolutionary diversity. So we have 10 years ago, we didn't have genomes of different fungi that we could tap into to ask which of these fungi can be good for our specific uses. So I think we go with these particular strains because we use, as scientists, a reductionist approach, right? We go into literature, we search for what's available, and then we start from there. However, if we take a reverse approach where we say, these are 6 million fungi, these are the genomes that are available to us that 10 years ago, whole genome sequencing has not caught up. Now we have all the genomes available of these fungi. Can we then go in and say, can we use computational biology and evolutionary perspective to see which genes are good for certain properties like foldable, bendable, fire resistant, does not give out odor, for example. So there's so much we can do from the biology perspective. That's why I think it's so exciting that when material scientists and evolutionary biologists and fungal biologists come together, we can dream about these questions that we could not have asked in the first place.
Josephine, if I could just add something, I want to just reinforce the point that the 4 or 5 species that we're tending to grow for biomaterials, we're growing because we were already growing them for other reasons. So Ganoderma is also known as reishi. We were growing that as a medicinal. Pluralis austratus is grown all over the world for food. And actually, interestingly enough, Tomasz, you might know this, but Pluralis austratus was first grown in Germany as food after a world war. So yeah, so we have not selected these fungi for their materials properties. We have selected them because they were already growing as pure cultures in the lab. And that's why I think Josephine's approach is really interesting and important.
If I may perhaps add here, this is a real shortcoming we have at the moment. As Josephine, you said, we use what we know, but we don't know if what we use is really the best for our purposes. And I remember there has been a work years ago, I tried to find it, about a mycelium somewhere in Southeast Asia, which is extraordinary stiff, where I was totally surprised. Maybe it's a measure error, but I have a look at, and I think really one could do several things. One could look at the real best fitting fungi for our purposes. And on the other hand, we have so many possibilities with genetic manipulations, because the materials the mycelium is made of is potentially mechanically very interesting. So it's, yeah, I think this is how we should tackle it probably, because at the moment we just use, as you said, the 5 or 10 fungi we know and the ones which are allowed to use without big safety issues. But in future we should really tackle it at least partly from this aspect.
Perfect. I just have a short comment to make there to Jen's point, and then I'll turn it back to Zoubeida Felecia as well, is that we use, so from a food perspective, we go through a safety regulatory process. So what that means is that we use strains that have been shown to be GRAS, generally recognized as safe. So while Jen is absolutely right that we select these strains because they work historically, but also because they're safe. So another aspect of fungi is like kind of the good, the bad, and the ugly. They produce some really nasty mold toxins, aflatoxins, which I work on, and many others. So absolutely, we use a handful of these strains because historically they've been proven to be safe for use. Zoubeida, I'm going to turn it back to you.
Okay, so I'd like to see Felecia. Did you want to add anything before we move on to the next question? Okay, all right. Let's move on to a related question from Zoom.
So there's a question again on fungi species. The question is, do you know if there are any fungi that can tolerate high salt content? Perhaps some which grow naturally on dead material in salt marshes. I think you already answered this question on Zoom chat, Josephine, but
yeah, I did answer this question.
Here.
Yes. So I'll just quickly make a comment that one of the beauties of fungi is fungal diversity. So if we look into, for example, high salt cheeses, we can select for a fungi that can tolerate high salts, right? They are extremely, they evolve resistance over time. Another beautiful ecosystem to look for them is in the marine ecosystem, where there is high salt concentration. So absolutely, genus that has been associated with marine sources.
Josephine, just a quick note that your Wi-Fi may be a bit unstable. I'm not sure if turning the video might help, although I love seeing your reactions, so that will not make that possible. So you make your choice.
I'll try to fix it. I'll
Okay.
try to fix it when Felecia talks.
Sure.
Thank you. Thank you for letting me know.
Okay, thank you. I did want to go to Felecia because we've talked about structure, right, and strains of fungi. And Felecia, in your introduction, you showed us beautiful examples of textiles. I would like to go to you and talk about it in terms of performance and processing. What are some of the ways in which textiles could be made with fungi? Maybe give us a little more specific, and again, with a focus on the processing aspect and the performance aspect that, as an expert, you're looking for.
I would say so. This is a new area for me, but I know that scientists and people are, especially in fashion design, because fashion design is another area where there are lots of cycles of textile use, and then quickly these textiles go into the landfill. So this has become a hugely pressing problem in our kind of clothing industry. But there are people working on different ways of using, for example, mycelium for a textile that you don't have to sew, you don't have to stitch it together. I believe earlier this morning, Elise showed a leather-like textile that she was working on. Other people are also working on wet spinning yarns out of mycelium to make biodegradable textiles. So I think that in our lab, one of the appealing things about these textiles is that they are biodegradable, because when you do make architecture with these materials, you are talking about a lot of material to shelter someone. They are indeed very lightweight. So that is one kind of reduction that's helpful. But when you talk about making a tent, a 10 by 10 area that gets covered by a tent, that's a lot of material. So having a material that can be biodegradable is really important.
All right, thank you. We talked a little bit about this. I'd like us to revisit it a little more in depth. Are these fungi safe for use as building materials? Tamás talked about regulations in Germany, and Felecia, you had comments about here in the US. Can we talk about it, making building materials a reality?
If I may say a word here, I think it would be tremendously important to cooperate with psychologists as we do with our living material systems, which make a thing which is called acceptance research. Because I think it might be a problem to convince people to use mycelium stabilized or built buildings, because fungi, as we all said, have this feeling of being potentially dangerous. And I think before doing this, I would really recommend we do this for our living material systems, because we don't know what's accepted by the public. And therefore we have a psychologist group in our LiveMats and they do this acceptance research with so-called cognitive affective maps. And it's surprising. To me, it's totally surprising what's accepted, sometimes which I would never accept myself, and what's not accepted where I would say, oh, good God, no problem at all. And I think especially with these fungi-based materials, one should do it relatively early, not to run into a direction where you develop stuff which is great, but nobody wants to have it.
Absolutely. I think this is part of my question also of getting it outside the lab. And trying to understand what are the kind of best ways of bringing it to a public outside of academia. You know, I was talking about earlier, you know, does it appear as a brick, or can we actually let the public in on part of the process so that there could be workshops and communities, and there could be more control over structures that get made outside of, you know, our purview at academic practice. And I think that that's a huge issue, because people in general, many people are afraid of fungi, right? Yep. They've often brought bad things, and we've spent a lot of the last 2 decades figuring out and making agents that get rid of them, right? So I think this is a huge issue to overcome.
If I may add here, if you have a chance, I would recommend to do what we always do, like this fiber-based building. If you have a botanic garden around, try to use a botanic garden as a kind of showcase place, because people going into the botanic garden, they are very open-minded and they accept more strange things than in normal environments. And if they could see, for example, that your wonderful kind of close fungi buildings work in a botanic garden and they are not killing them, this might really help to lower the level of acceptance, so that they say, oh wow, why not? I don't know if in Penn State there is a botanic garden, but this is normally a very good issue to get people, visitors, accepting things which they wouldn't accept outside.
Yes, we have an arboretum here. Perhaps there could be workshops, right? Connecting with populations. I think Esther was talking about this earlier. Populations who do not normally have access to thinking about building materials like this. So.
Yeah, Josephine, would you like to add to that?
Sure, I'll keep it short. So in case it cuts off, it's not kind of annoying. I know even after 2 years of Zoom, this still happens. So I think when we think about the food space with, you know, funky cheeses, right? You know, stinky fungal foods. The best way to do it is to partner with chefs, right? To bring these products out into the market, to have people try them. So Thomas and Felecia brings up a really great point about social acceptance. We need to partner with folks that are on the other end of the spectrum, right? Like with chefs, for example. When I think of food, people need to taste these things or feel them in this case. Don't taste fungal bricks, but you know, taste these, touch them, feel them, kind of be more open to share what these materials can do for us. So I think that's the only thing I would add there is sort of the science outreach and communication part of this.
Great. I'd like to pause and see if there are any questions here. Yes. Okay. We're going to take a question from the in-person audience. Do you want to introduce yourself?
Hi, panelists. My name is Niroshan. I'm a postdoc from biological department. So my question is, you said the fungal material is like relatively weaker, right? So I'm just wondering like what are the processing techniques we have to increase the strongability or like rigidity of the fungal material. The reason that I ask, like if you take like a A5 one paper, that is like very soft, right? But if you press like 100 papers, it became like very rigid. And the second question is, if we make like a very applicable fungus, like a fungi material for the like a building material, so what would be the durability of that material? So I'm thinking the like if they don't like, if the durability is like very low, then people will not buy it, right? So like I'm if you give some idea about that, it's really appreciated. Thanks.
I can answer the first question, and then I'll probably turn it to the rest of the panelists for the building question. So Niroshan, your first question on growth conditions, right? We see these, I'm not sure if anyone has had turkey bacon. That's a food product where essentially the protein is pressed in layers. So absolutely, the growth conditions can really drive mechanical strength. And I think MicroWorks is leading some of these proprietary growth conditions. How do you grow the fungi in order to select for these high mechanical properties? The other way is we can grow fungi on solid medium as well as liquid air interface. So there's a lot of technology and intellectual property going into how do we grow these fungi to optimize these growth conditions to select for biomass. Again, the last point I'll make is on the genetics itself. We know that there is a protein in the cell wall of fungi known as hydrophobins. And when you take that protein out, fungi lose that mechanical strength, right? So how can we increase the expression of hydrophobins, for example, and select for these mechanical properties?
Would anybody like to address the question about durability?
Perhaps one aspect, I think one has to keep in mind that the matrix material on which the fungi grow, if it has a stabilizing impact on the building, should not be eaten up by the fungi. I think this is sometimes not so easy because a lot of these fungi can feed on many different biological material, bio-based materials. So I think this is also important to keep in mind. I don't know, Felecia, what kind of clothing you use. Is it cotton or sisal or whatever?
That particular model that you saw was linen, but we're running through a gamut of different yarns and substructures.
Have you seen that your fungi feed on the clothes, or?
Actually, we have not seen them. We're right in the middle of our, I guess you could say we're right in the middle of our experiment.
Yeah.
And we are looking for that. We're looking to see what it likes to digest. We also have some other yarns that we wanted to try. There's one called algae knit made from seaweed. There is another yarn made with orange peel. So we're trying lots of different things on small samples right now as well. So we should probably talk about that more, Josephine, as well, to see what the fungus likes to eat and digest and what makes a kind of strong root structure for, and also what happens to the substructure when you put it under stress and strain, right?
Yeah, hi, I have a question, and also I just wanted to show one slide just to inform. So this is from a 2019 paper, and it's a very complicated figure, but if you just look at the biggest rectangle, that is the currently described species diversity. Now we're not talking about strains, we're talking about species, which are complexes of strains. And you see a couple of big divisions. Ascomycetes are in green, and the basidiomycetes are in blue. And I know those are jargon terms, but it's worthwhile to know something about them because ascomycetes, for example, are the ones that produce aflatoxins and other things. Basidiomycetes in the blue are typically the ones that we're using in fungal biomaterials because of their cell wall properties. And there are lots and lots of people who know way more about this than I do, but I just wanted to put up this picture that even among these safe, thick cell-walled species, we're growing 10 or whatever, probably more if you include cheese. But there's 48,405 described species, and many, many more that we have not described. So I just wanted to drill home the point that we really have not scratched the surface in terms of potential for building materials. And then, thank you, Vinay. Then I also I wanted to just say that historically there's been two currents in human history with relation to fungi, and one of those is mycophilia, and one of those is mycophobia. And I'd want us to keep talking about those two threads because I think they're both very important historically, and I think the idea that's been raised that we should, we should have some focus groups, we should have some expos in botanical gardens, we should figure out how we're going to make fungi palatable as building material. The social aspect is really important. I guess those were not really questions, sorry.
Any comments from or reactions to what Jen just shared? Yes.
I may say one thing which was very interesting. There was an exhibition 2 or 3 years ago about the rhizosphere in woods, in forests. And it was really interesting. You could walk underneath in a kind of tunnel and you see these fungus infected roots of the trees, and everybody was, it was not in our garden, it was another garden, and everybody, they got the feedback by the visitors, was totally enthusiastic. I think it's partly how you present it. You have to give it a kind of good, supportive, future-bound background. And there was the saying, okay, we need the fungi, the rhizospheres that our trees survive even in coming drought, and so on and so on. And I think this is really part of the message we have to sell if we want to establish this fungus stabilized or inspired architecture, said it's a good thing and nothing potentially dangerous.
And I will just add here. Oh, sorry, Felecia, go ahead. No,
Felecia. You go ahead.
All right, I just wanted to give a shout out to my colleagues in plant science and plant pathology and environmental microbiology. So our colleagues over there have been working with plants and fungi for a long time, right? So I think the tools are there to leverage. We just need to bring them into the conversation. So now we can use these existing tools that are not new and apply them into tapping into fungal diversity for bio-based materials.
Josephine, you are hard to understand again.
We lost you, Josephine.
Yeah.
Thank you for letting me know. Sorry, can you hear me now?
Yeah, now it's better. Can you repeat your last part?
Yeah, sure. I will repeat it. Thank you, Thomas. I apologize. Thank you for your patience.
No problem.
So I wanted to say that just a shout out to my colleagues in plant science and plant pathology, because they've been working with plant fungal interactions for a long time. So we can repurpose some of those tools, leverage them, and apply them to biomaterials.
Mm-hmm. Yes, that's true. Yeah.
Felecia, did you have anything to add as well?
I was just thinking about the phobia aspect, right? That many people have, and I think that I'm also working on a project that's looking at ways in which people engage STEM topics in schools, and I think that potentially this is, an area that the National Science Foundation might kind of tap into, that it could be a grant that one goes and starts to introduce workshops to people, because I do believe that these phobias and also philias start really young, right? Really, really early. And so setting up a series of workshops or talks or something that travels around throughout the country would really change people's attitudes towards nature, right? And using nature for building products and wearing them and including more of these species into our lives.
Yeah, that's a great point, Felecia. And in fact, as we mentioned this morning, one opportunity is from the National Science Foundation Emerging Frontiers in Research and Innovations, the NSF-EFRI program, which when it talks about bio-based materials in the built environment, also talks about social acceptance, social aspect, outreach to the general public. And it specifically mentions that it is not looking for outreach opportunities, as Esther mentioned earlier, right? Where we develop the technology, then we go out and say, look, isn't it awesome? They're looking for those aspects to be integrated in concert as the research questions are being developed. So definitely opportunities, and we have opportunities tomorrow afternoon to talk about all this and how we can address it in an interdisciplinary manner. Yep. We're gonna go back to Zoom for a couple of more questions.
There's a question, right, about the social acceptance, actually. So the question is, does the panel have any thought about how researchers can engage farmers and growers to ensure that these materials can be scaled with the needed support from these growers in the near future? I don't know if this is the right panel, but this could be the next panel, a question that we can ask for the next panelist. But maybe, Josephine, do you have any ideas about this?
And how, if I may repeat the question, so the question is, how can we engage existing growers and farmers to grow these new materials?
To ensure that these materials can be scaled with the needed support from the growers in the near future.
Yeah.
The
great question. So I think one of the land grant missions of Penn State is its extension mission, and we already have an existing network of farmers and growers, and Pennsylvania is the largest for mushroom production. So I think a lot of what we can do is to tap into existing resources and reskill our existing workforce to build these new. So I think the first step is engaging them, and then how do we have the resources to reskill existing workforce? I hope that answers the question. Sorry, it's a little hand wavy. I don't have any data or anything on that front.
This question, Josephine and Felecia and Tamas, relates to what we discussed too, right? We wanted to bring up, which is the availability in large quantities and low cost, and what the outlook is for both in the next few years. So maybe we can address it in that way as well.
I can say very little to this because I have to admit that what we do normally, we hand over if we have a kind of demonstrator, a prototype like thing. So we are not collaborating a lot with farmers or mushroom growers, and that's why I have to say I can't say anything, but I know from Germany that there is, like, and probably like in the States, there are more and more of these persons who grow commercially mushrooms, and I think there is enough manpower, woman power around to do it, and also the will to do it, but I have no idea how it looks like in the States in this aspect.
And I think it also goes with demand, right? Like I think the supply and demand, if there is a demand, then there would be an economy for it. And I think about it a lot from a food perspective or from agricultural perspective as well. It took us a long time to domesticate corn, for example, right? Like the food that we eat. And I think the same thing, if this is sort of the future, how do we incentivize to give farmers the incentive to grow these materials? Felecia, I'll turn it to you. Sorry if you jumped in there.
Yeah, jump in. I would say, coming from architecture, where there's much less research money, that relatively speaking, if you're talking about the kind of research, setting up the research agenda, that working with this material has been relatively speaking low cost. And I'll compare it to, for example, another research project that we have been involved on, working with a solar fiber, which was hugely expensive, right? Just stratosphere costs in terms of chemicals and getting the chemicals onto the fiber. And at the end, you know, we could work with one fiber. So I think that in terms of experimentation, what's really nice about this particular area with Mycelium is that it seems to be relatively low cost and that people could access it fairly easily. In other words, when we talk about bringing it out of the lab, I suspect that it would be possible to have workshops with a community, and we would be able to afford research with others, right? Working as a kind of collective. What happens when you think about constructing something as a group as opposed to individually? So I think that those things are really a pleasant surprise about these materials.
If I may add here, I think we may ask even the wrong question. Would we really need huge building blocks prefabricated? Wouldn't it be for architecture not much better if you build the scaffolds of the materials and then infest the scaffolds with the fungi mycelium? So it would not be necessary to have farmers or so who build large building blocks. So the architects or the developers or whoever builds a building out of a suitable matrix, and then it's infested by the suitable mycelium. And I think this would be much, in my opinion, would be a much better way to do it.
Yes, I like the term infestation.
Yeah.
I have a question for Tomas about the presentation. We didn't have a long Q&A session after your presentation. I wonder if you or your research group also looked into not the fungi as a biomaterial, but the growth mechanisms of different fungi species. And did you learn any? I don't know if you looked into it, or if there are any plans to look into those.
We looked at. I have to say, I always look at fungi when I grow my plants in my greenhouses, because without fungi, no plants. So the fungi are always, let me say, collaborators for us. But I was interested in some fungi years ago, because you have some fungi which fill exactly all space which is available. So they have no predefined form, some of the fruit bodies. And this was interesting to me, but we never looked into detail in it, because I have to admit, in Germany, the regulations to work with fungi, as I said, are so complicated that we would have to set up as one lab only for working with the fungi. And I said, for my sheer interest, which was more or less the interest of the boss of the institute, I wouldn't set up as one lab. So, but there is definitely a lot to do. And also what I like very much, because some of the fungi are extraordinary, the fruit bodies are extraordinary fast growing. And this could be also interest, because if we look at fast growing plants also, we always look at bamboo, but compared with some fungi, bamboo is slow. But perhaps Josephine can add something on that, because I think fungi are tremendously producing biomass in a tremendous speed sometimes. Or?
Correct. Yeah, and that is why we use Rhizopus oligosporus as a model for a fast-growing fungi. It's a food fungi, but it tells us a lot about their biology. We can grow biomass in 24 hours, pretty, as a sheet. And the reason why we use it in food is because it's fast-growing. So I think that's the other reason.
Thank you very much. Any last question from the audience? Okay, thank you. I think we heard a lot of, first of all, great information, and also the information that we heard. Are you waving at us? Hi.
My students are in the audience, and I think plant pathology's department head is sitting in the audience too.
Yes,
so I'm
yes. And so what I was going to say is, in addition to the excellent information we heard, I heard multiple times that many of these, what I would call opportunities, are going to really require interdisciplinarity and multiple toolkits and multiple approaches in order for us to address them. And so I'm for one elated, because this is, around fungal biomaterials and biofabrication, we can again put together multiple teams that address applications, processing, material based approaches that maybe will help us in figuring out spaces to do this research safely, but also really help us develop the knowledge around this in a faster way, like bamboo and fungi, than it would, if we were sort of siloed, which a lot of the times we are in our own particular areas. So thank you very much. Thank you, Felecia. Thank you, Josephine. Thank you, Tamás, for your contribution.
Thank you for the invitation, and I will have a walk at the beach. See you
yes,
soon.
I know. Rub it in. I know you're in
Thank
Hawaii.
you.
Enjoy Hawaii.
Yeah, I definitely will. Definitely will. So see you soon.
Bye-bye.